Hello everyone. Let's see about ligases, linkers and adapters. First we'll see about ligases. DNA ligases catalyzes the formation of phosphodiester bond between two deoxynucleotide residues of two DNA strands. DNA ligase enzyme requires a free hydroxyl group at the T-end of one DNA chain and a phosphate group at the 5' end of the other and requires energy in the process. E. coli and other bacterial DNA ligases use NAD+ as energy donor, whereas in T4 bacteriophage, T4 DNA ligase use ATP as cofactor. The role of DNA ligase is to seal nicks in the backbone of double-stranded DNA after lagging strand formation to join the Okazaki fragment. This joining process is essential for the normal synthesis of DNA and for repairing damaged DNA. It has been exploited by genetic engineers to join DNA chains to form recombinant DNA molecules. Usually, single-stranded breaks are repaired using the complementary strand as this template, but sometimes double-stranded breaks can also be repaired with the help of DNA ligase 4. The most widely used DNA ligase is isolated from T4 bacteriophage. T4 DNA ligase need ATP as cofactor. The enzyme from E. coli use cofactor NAD. Except this, the catalysis mechanism is somewhat similar for both the ligases. The role of cofactor is splitting and forming an enzyme AMP complex, which further aids in formation of phosphodiester bonds between hydroxyl and phosphate groups by exposing them. Mechanism of action of DNA ligases. ATP or NAD+ reacts with the ligase enzyme to form a covalent enzyme AMP complex in which the AMP is linked to amino group of a lysine residue in the active site of the enzyme through a phosphamide bond. The AMP moiety activates a phosphate group at the 5' end of the DNA molecule to be joined. It is called as the donor. The final step is a nucleophilic attack by the 3' hydroxyl group on this activated phosphorus atom, which acts as a receptor. A phosphodiester bond is formed and AMP is released. The reaction is driven by the hydrolysis of the pyrophosphate released during the formation of the enzyme adenylate complex. Two high-energy phosphate bonds are spent in forming a phosphodiester bond in the DNA backbone with ATP serving as energy source. The temperature optimum for T4 DNA ligase mediated ligation in vitro is 16 degrees Celsius. However, ligation is also achieved by incubation at 4 degrees Celsius, by incubating overnight, or at room temperature condition by incubating for 30 minutes. Adenylate and DNA adenylate are the important intermediates of the phosphodiester bond forming pathway. Ligation efficiency depends on the ends of the DNA in the reaction. Mainly two types of ends. One is sticky end, another one is blunt ends. In the sticky ends, ligation is efficient. Annealing of complementary overhands brings 5' phosphate and 3' OH into close proximity. In the blunt end, ligation is less efficient, need high concentrations of ligase and DNA. In blunt end ligation, mainly three methods can be used to put the correct sticky ends onto the DNA fragments. First is cloning foreign DNA by adding linkers. Second one is cloning foreign DNA by adding adapters. And the third is homopolymer tail adding by using terminal transferase enzyme. Next we will see about linkers. Linkers are short pieces of double-stranded DNA of no nucleotide sequence that are synthesized in the test tubes, that is synthetic oligonucleotide. These are blunt ends but contain a restriction site. DNA ligase can attach linkers to the end of large blunt-ended DNA molecules. In a blunt end ligation, this particular reaction can be performed very efficiently because linkers can be made in very large amounts and added into the ligation mixture at high concentration. More than one linkers will attach to each end of the DNA molecule, producing the chain structure. However, digestion with BAM-H1 cleaves the chains at the recognition sequence. producing a large number of cleaved linkers and the original DNA fragment, now carrying BAM-H1 sticky ends. This modified fragment is ready for ligation into a cloning vector restricted with BAM-H1. Next, we'll see about adapters. Adapters, like linkers, are short synthetic oligonucleotides. But unlike linkers, an adapter is synthesized so that it already has one sticky end. Using adapters has one more problem. The sticky ends of individual adapter molecules could base pair with each other to form dimers. This leaves the new DNA molecule still blunt-ended. The sticky ends could be recreated by digestion with a restriction endonuclease, but that would defeat the purpose of using adapters in the first place. To solve this problem, we remove the phosphate group from the 5' position of sticky ends of the adapters by the help of alkaline phosphatase enzyme. Due to this, DNA ligase is now unable to form a phosphodiester bridge between 5'OH and 3'OH ends. The result of this manipulation is that although base pairing always occurs between the sticky ends of adapter molecules, the association is never stabilized between them by ligation. Adapters can therefore be ligated to a blend-ended DNA molecule but not to themselves. After the adapters have been attached, the abnormal 5'OH terminus is converted to the natural P form by treatment with the enzyme polynucleotide kinase, producing a sticky end fragment that can be inserted into an appropriate vector. They are also short double-stranded oligonucleotides that carry an internal restriction sites and single-stranded tails at one or both ends. This protruding sequence can be ligated to DNA fragments containing a complementary single-stranded terminus. After ligation, the DNA can be cleaved with appropriate restriction enzymes to create new protruding terminus. Adapters are available in two basic designs and a variety of specifications. Some consist of a partial duplex formed between two oligonucleotides of different length. For example, EcoR1 NOT1 adapter. Another class of adapter is supplied as an unphosphorylated single nucleotide whose sequence is partially self-complementary. As example, EcoR1 PST1 adapter. Next, we will see about homopolymer tailing. It is a technique which sticky ends can be produced on a blunt-ended DNA molecule. In a homopolymer, all the subunits are same. A DNA strand made up entirely of deoxyguanosine is an example of homopolymer and is referred to as polydeoxyguanosine or poly-DG. Tailing involves using the enzyme terminal deoxynucleotide transferase to add a series of nucleotides onto the 3'OH termini of a double-stranded DNA molecule. The reaction, when carried out in the presence of just one nucleotide, then a homopolymer tail will be produced. This picture depicts the formation of homopolymer tailing. For ligation of two-tailed molecule, the homopolymers must be complementary. Frequently, poly-DC tails are attached to the vectors and poly-DG to the DNA to be cloned. Thank you for watching. Have a nice day.